we start today cell function. Actually, cell function comprises of different areas. Number one is osmosis. What is osmosis? Whenever two solutions of dissimilar strength have been kept separated from each other by a perfect semi-permeable membrane, the process by which solvent molecules move from low concentration of the solution towards the high concentration of the solution until both of the concentrations are reaching the equilibrium. So this is what is osmosis. Now what is diffusion? If you ask me, the term diffusion means when two solutions are kept separated from each other, the process by which bulk movement of molecules occur from high concentration to low concentration until both of them are reaching the equilibrium. This is what is diffusion simply. So better you can say that osmosis and diffusion, they are very much interrelated, and we can use a language, specific terminology, that osmosis is a special type of diffusion. Why we say so? Before we go into the detail of the concept, we must be using three specific terminologies. What is meant by isotonic, hypotonic, and hypertonic solution? Whenever the extracellular concentration is equal to the intracellular concentration, the language is isotonic. So if somebody asks you what is hypotonic, then obviously you can say the intracellular concentration is less than extracellular concentration. Then what is hypertonic? You get just the reverse. That means intracellular concentration is greater than extracellular concentration. And you can say that the concentrational difference will do two things. In isotonic, they will never create any problem because the two concentrations of two sides, they are equal, so cell will never show any structural deformity. But in relation to hypotonic and hypertonic, I can suggest two things. If it is hypotonic, then obviously the cell is immersed in outer dilute media and the cell will gain more water as a result of endosmosis. So as a result of endosmosis, it can be said as the outer one is more dilute, water will logically come inside it and the cell will swell and may ultimately lead to bursting. Coming to the concept of exosmosis. Whenever a cell is kept in outer hypertonic solution, the process by which water molecules go out from the cell as a result of exosmosis, the cell will obviously shrink. This is what is the peculiarity of exosmosis. So, on the basis of these two things, it can also be stated that there are two very important conceptual languages. One is plasmolysis and the other goes to deplasmolysis. So if somebody asks you what is plasmolysis, Whenever a plant cell, this is a specific language for plant cells. Whenever a plant cell is kept immersed in outer hypertonic solution, the process by which shrinkage of protoplast takes place. So, plasmolysis is definitely exhibiting a shrinkage of protoplast. On the other hand, if the already plasmolysed cell is kept immersed in outer hypotonic solution, there is a recovery of turgidity, and this is what is the deplasmolysis. So, considering these languages, if we come back to our original question, that osmosis is a spatial diffusion. It is nothing but a diffusion of free solvent molecules from high free solvent zone to low free solvent zone. It means movement of solvent from low concentration towards high concentration, obviously, until they attain equilibrium. Thank you very much. For testing your understanding of this lesson and more videos, log on to www.tubelessons.net.